A bulk transcriptome from acute leukemia usually consists of thousands of cells, and these cells creates the subtypes from these acute leukemias. And in this project, I actually checked whether these types can be estimated by the cell composition of these, or was there another cell intrinsic effect that we cannot really predict. I did it in 4 parts. I built a single cell reference, and I downloaded matching samples, single cell RNA sequencing and RNA sequencing, to select a cell estimation method. And then I created a bigger cohort for bulk RNA sequences across 3 different diseases, acute leukemia. And then I tried to split this cell composition versus cell intrinsic signal. First, I created this UMAP plot, which consists of a bone marrow atlas plus a thymic atlas. I added the thymic atlas because TALs are created cells from thymus, and the bone marrow atlas actually lacks the cell. I did those to cover the whole myeloid and lymphoid lineage. And then I checked this 16 samples on bulk deconvolute levels versus actual single cell data, and then I found out the Instoprism turned out to be the best method to deconvolute these samples. And then I deconvoluted these 700 or so samples, regrouped them based on their subtypes to see whether we can discern the cell composition that I expected from these different disease status. And surely I got more myeloid lineage cells for acute myeloid leukemia, and B cell types for BALL, and T cell type for TALL. And when I did a normalization across cell types, I saw more rare cell types to pop up, and you can actually discern more about the subtypes using the deconvolution itself. And then I trained linear models to whether I can predict the subtype using these 35 estimated cell compositions. And I trained another model with these 35 cell compositions plus 15 genes that are specific to that subtype. So if these 15 genes actually add any value to the model itself as cell intrinsic component, and big aberrations like translocations like PMR-RARA or NX1T1, I found out cell composition explain most of the predictability. Whereas if I go to smaller changes, SNPs, then I found cell intrinsic effect starts to take off. And I wrote a lot about it. I wrote a blog post here and more of the analysis that I've done, and you can check it out from this website and then see what I've done in detail and more. Thank you for listening.